Good morning, guys. This is your host, Zarbuft, with the Brain and Health Talks at SABCR. Our Canadian weather is acting up a little, and we've already had 2 heavy snow days, but the show must go on. Today with me is a very special guest who is involved in cutting-edge research in understanding challenging neurological diseases. His work is so unique in its core that his own novel technique could potentially provide answers for neurological diseases that have genetic bases. I'm very excited to have Glenn Walpole with us. Glenn, could you please elaborate in layman terms, as we do have people tuning in from different parts of the world, and some of them could be very young to understand scientific terminology. Could you please explain to them the wonderful work you're doing at McMaster University?
So first of all, I'm thrilled to be doing this interview. So I work in the laboratory of Dr. Ray Truant here at McMaster in the hospital, the Health Sciences Center. So we focus on Huntington's disease, which is a genetic disease that leads to neurodegeneration. Now, when you think of neurodegeneration, you might have Alzheimer's or Parkinson's or maybe Huntington's, ALS. There's a lot of different diseases that come to mind. And we study Huntington's disease for a very particular reason, and that's that it's purely genetic. It's carried through families in an autosomal dominant fashion. So what that means, if you've never heard of that, is that it only requires one faulty copy in your genome to carry it through. So if your mother has Huntington's disease and she passes on that faulty copy, you will have the disease.
And it's not something that can make the susceptibility increase or decrease. It's just that if you're genetically predisposed, you're sort of like, it's a sentence.
Right, you have the disease if you carry the faulty copy. So it's an interesting disease because the age of onset is actually quite late. There are cases where it's early, as in 20 years of age, but it tends to be around 40 to 45 years of age when people start experiencing symptoms.
So it's mostly the elderly populations or middle age?
It's mainly around 40 years of age. And you'll actually hear about stories where people go from having completely normal lives. They're very successful, working in high-end companies, going about their normal activities, and then everything changes, and it ends up that they'll be diagnosed with Huntington's disease, and they otherwise had no idea.
It does affect people's lives significantly, even on a psychological, emotional, and a physical level.
Right, so it's affecting many things. This disease not only affects the brain, but it spreads through the body. So the main symptoms would be chorea. That's the official name for it, but that's actually, it refers to kind of rapid, uncontrolled motor movements. So these people lose the ability to control their limbs, basically. It's common to have depression if you have Huntington's disease. There's significant intellectual decline. And of course, once the symptoms start, if that's around age 40, it's usually 10 to 15 years before that person passes away.
And that reality is so closely replicated with brain tumors and brain cancer, and a lot of neurological diseases sort of intertwined.
Right. So our lab, we focus on Huntington's disease for a specific reason, and that's the genetic reason. Sometimes you hear about Alzheimer's or Parkinson's, but it's actually quite hard to describe exactly what that is. There are genetic components to some of those diseases, and then it's referred to as a familial form. But Huntington's disease is purely genetic. So in terms of that, it's a lot easier to look at the mechanisms involved in the disease.
So Glenn, could you discuss with our listeners the prevalence of this neurological disease?
Sure. So this is actually quite rare, the Huntington's disease. So it ranges from about 5 to 10 per 100,000, and that's from in a North American population. So it's not so much that it's affecting so many people, but it's an important disease to understand not only this specific disease, but many other neurodegenerative diseases.
And that's a very significant number.
Right. Now, when you actually look at when they discovered this disease and the actual gene that codes for this faulty protein, they actually studied a small population in like a very remote village, because if you think about it, if it's a small population, the gene, that faulty gene, will be very common in that population.
It'll get replicated.
Right. So these people were referred to as having the shakes, and that refers to the chorea, very uncontrolled motor movements that you experience with Huntington's disease.
So this neurological disease, we keep hearing the word that it has a genetic basis. What exactly do you mean that it's a genetic disease?
Okay, so shown on this diagram on the slide that you're looking at, this is a representative image of the Huntington protein. So when you say that it's genetic, in all organisms, genes code for proteins. So this gene that has a problem with it codes for a protein that's basically a faulty protein. So as you can see in the image, you can break up proteins into many different segments. And in Huntington's disease, there's one specific part of this protein represented in the blue square that's basically too big. It's expanded.
So you mentioned the expansion of the protein. What does that mean? And could you also explain what the expansion does to the protein?
Right, okay, so in Huntington's disease, that gene codes for a protein called Huntington. It's very well named.
Mm-hmm.
So if you look on the slide, in a healthy individual, on the left, you can see that blue box, which I talked about on the previous slide. That represents the region that gets expanded in the disease. So proteins are made up of building blocks referred to as amino acids. And there's a particular amino acid that is in this expansion, and that's glutamine. So in a healthy individual, there's basically less than 36 glutamines, or 36 Qs, as you can see on the picture. In the disease form, someone with Huntington's disease will have more than 37 Qs or glutamines. So that's why we say it's a glutamine expansion. So what does this expansion do? I like to use the analogy of the protein being like a door hinge. So in the healthy door hinge, you have a brand new hinge that swings back and forth very freely with no friction. And that's a lot like what the Huntington protein is like. So as you can see on the image, what I'll refer to as one side of the hinge is the N17 on the diagram. The other side of the hinge is the poly P. Now, if a hinge can move back and forth very freely, but when you actually go to the disease form of the protein, it's like a rusty hinge. So you left your door outside too long, didn't have rust protectant, and
didn't use the spray, and now it's too late.
Right. So in Huntington's disease, the beginning of the protein is kind of like an open rusty hinge. So the N17 shown on the diagram, it can't even fold at all. It can't get back to the poly P on the diagram. So normally a healthy protein, it's just like the good hinge where it can open and close very dynamically, and this is lost in the Huntington's disease protein.
So Glenn, could you describe this protein in magnitude? Like what does it really even look like?
So you can't actually see the protein when you're working with it in the lab, but if I could describe the actual size, you can imagine the little diagram on your screen is actually only a very small section of the protein, and the full protein, if I could represent it on your computer screen, would have to go right outside of your house. It's a huge protein, so this is only a very small section of the actual protein.
Wow.
Right? Yeah, it's a humongous protein.
Wow, and we're only looking at a very, very small segment of it.
Right.
So what does the rusty hypothesis have to do with the disease itself?
So on the slide, you can see that there's a bunch of different little dots, and what these dots are representing is the actual protein. So if you imagine the hinge that's working properly, that's the small little dots on the left. They're folded correctly in the biochemistry terms. So they're on their own, they're flying around in the cell doing what they need to do. Now if you add in too many glutamines and cause that expansion, you get the rusty hinge, and that is a misfolded protein. So that starts to lead to what we call protein aggregation. So as you can see on the right, it's almost like the protein is like Play-Doh. It's sticking together, forming these huge clumps that form balls or aggregates in the cells. So this is a problem because if you think about the protein, it normally has many different functions in the cell. But if you're throwing it into this big ball of Play-Doh, it basically can't get around the cell to perform the functions it needs. And these aggregates of protein are actually common to many neurodegenerative diseases, such as Alzheimer's and Parkinson's.
So you raise a very interesting point in terms of protein aggregation, and I'm wondering if there is a common trend when it comes to neurodegeneration.
So as you can see on the slide, these protein aggregates are actually common to many neurodegenerative diseases. So we have an example of Huntington's disease on the left in A, Parkinson's disease in the middle, and Alzheimer's disease on the right. So they're all protein aggregates, but it's very complicated in terms of deciding whether these aggregates are actually a good or bad thing. You might assume they're a bad thing because it's not found in normal people, but at the same time it actually might be trying to compensate for the misfolded protein. So in just Huntington's disease alone, we've actually seen many examples where papers and different labs around the world suggest that these protein aggregates are actually toxic to the cell. And then at the same time, other labs around the world will say that it's actually a protective aggregate. So one might say that when the misfolded protein is moving around the cell, that's when it causes the problems. And the other side of the story says that when it's actually in the aggregate, that's the problem. So maybe the huge aggregate in the cell is blocking movement of different proteins that should be able to get through the cell, but this aggregate is just acting as like a huge roadblock in the way.
So Glenn, those are two very different perspectives and viewpoints, very the opposite end of the scale. How do you even form a consensus on these two polar views?
So it's a very difficult point, but actually just this year our lab published another paper addressing possibly this exact question of why there can be two totally different ideas of what these protein aggregates are doing in the disease. And what they found in this paper is that there's actually two different types of protein aggregates that can form. And as you can see in the image on your slide, there's a depiction of what the protein looks like at the top, and then it can aggregate into the actual inclusions, and there's two different types. There's a very tightly packed inclusion on the right, and then a less tightly packed inclusion that's larger on the left. So these two different types of protein aggregates or inclusions have very different properties. The one on the right, called fibrillar inclusions, doesn't really exchange with the environment around it in the cell. So it's like it takes up Huntington that's misfolded and it packs it in very tightly and doesn't exchange it. The one on the left, it takes up Huntington, but it also exchanges it with the environment fairly readily. So you might suggest, and this wasn't really addressed in the paper because it's definitely a future topic of study, but maybe one is a safe aggregate and one could be the toxic aggregate.
So when you're studying all those intricate proteins and molecules and biomolecular mechanisms, how do you do that? How do you keep up with that?
So an extremely useful technique in terms of cell biology and biochemistry came about in a very odd manner. A supervisor in Asia was walking home from work one day and he looked over to the harbor and saw jellyfish, saw them fluorescing.
All things
right.
Jellyfish.
So good scientists, when they see something, they take advantage of it. They try and understand that system. And so this scientist saw these jellyfish fluorescing and said, I wonder what makes them fluoresce. And it turns out that that was probably the biggest discovery, and it led to a Nobel Prize for that individual.
Wow.
So what he actually found is that it's a specific protein, and a variant of it is called the green fluorescent protein shown on the left, top left. So what you can do is actually attach this green fluorescent protein to any protein in the cell that you're studying. So in our case, we would attach it to Huntington as an example, and then you can follow Huntington in cells.
And that's a very useful technique.
Right, so then you can use it watching Huntington in the cells. You can change the system. So say you're looking for new drugs. You can add the drug to your culture of cells and see what that does to Huntington. Does it make it go to the nucleus, for example, or does it form aggregates, or what does that do?
And what possible solutions we can come up with for future mechanisms.
Exactly.
That's so very important.
Right.
So what about that image on the bottom left?
Okay, so the scientist who discovered the green fluorescent protein, he was kind of showing off in a way. So what he did is actually derive different colors of this fluorescent protein. So you could have green fluorescent protein, a red color of fluorescent protein, blues, any color you want. And he streaked them on a plate in bacteria and basically depicted a scenery of a sunset.
Wow. Whoa, so that's a picture with the bacteria and colorful.
Right, so the bacteria are actually fluorescing because they're expressing this protein in the bacteria. It was just for kicks.
So, given all of that fascinating information, what is your specific area of research within this realm of different ideas?
So I work with a protein called PAXin1, and that's listed and represented by the yellow-shaped moon on your screen. So PAXin1 is involved in Huntington's disease because it interacts with the Huntington protein. So as you've seen before, we have represented on the bottom of the slide the Huntington protein, or at least the beginning of the Huntington protein. Now PAXin1, we think, kind of sits down on the Huntington protein and interacts at 2 points, the N17 and farther downstream in the proline-rich region.
So moving on into the novel approach where this whole hype has been around, could you explain and introduce to our listeners the new approach that you've taken on?
Sure. So with PAXin1, if you put, for example, the full green fluorescent protein on PAXin1, it doesn't appear to localize properly in live cells. So what I did is I took a fragment, a very small fragment of that green fluorescent protein. So on the slide, you can see that PAXin1 at the top is attached to a very small yellow portion of the green fluorescent protein. And then you add the rest of the green fluorescent protein, shown at the bottom left corner, into the cells. And these two different chunks of the protein actually come together and form the original green fluorescent protein. So on this slide, I've shown a few images that I've created this year. So here I'm expressing these two different fragments of the green fluorescent protein attached to my protein of interest, which is PAXin1. On the top.
And what you were looking at before, Glenn is actually a very modest person, and he hasn't elaborated on how the pictures you're looking at is actually a work of weeks and weeks and months of research, and now we can actually see the fruits and outcome of his hard work.
I will say that research is a slow-going process at times. So as you can see on the last slide here, the top left two images are this PAXin1 protein with these two different fragments of the green fluorescent protein coming together to form the fluorophore, and it shows that green color characteristic of the green fluorescent protein. So how can we be sure that PAXin1 with the green fluorescent protein is actually localizing where we want it to in the cells, or we would expect it to? So we actually compare it to another cell on the far right, top right, where I've used an antibody that actually recognizes my protein of interest. So if you can imagine, an antibody recognizes PAXin1, it goes to wherever PAXin1 is localized in the cell, and you can actually make the antibody fluoresce, which produces another type of image. So we're comparing images, the left two images to the far right image.
So what does the future hold for the type of research that is currently being conducted?
So we believe in our lab that the localization of PAXin1 is much improved with this technique. In terms of the future, we can do a lot of studies from understanding where PAXin1 is localized in the cell, because it's very important for Huntington's disease. So I mean, there's multiple avenues that I'll be taking this summer and next year to understand how PAXin1 plays into Huntington's disease.
So the research that is being done is vitally important and will be providing some important answers in years to come. Thank you to our special guest again, and to everyone who tuned in for our talk today. We will see you soon.